we've talked a lot about Boston Children's and Dr. Kleefscher's clinic in the Netherlands. It's important to remember there's other people who are working on Kleefscher syndrome. One of those people is Jacqui Harris. She's seen a lot of Kleefscher patients at her clinic at Kennedy Krieger at Johns Hopkins University. And she also has done a lot of work around clinical trial readiness in other conditions, but conditions that are disorders of the what we call the epigenetic machinery, which Kleefscher syndrome is, without going too deep into the science, Kleefscher syndrome is one of those conditions. So we asked Jacqui to come and give us her perspective on clinical trial readiness. Um, and I'll let her introduce herself beyond that.
Hi, thanks, Eric, for that introduction. Can everybody hear me okay?
Yeah,
Okay, great.
coming through fine.
Okay, perfect. Yeah, so I am Jacqui Harris, and I see patients at Kennedy Krieger and Johns Hopkins here in Baltimore. And I have a number of patients with Kleefscher syndrome because primarily what I see are patients with, as Eric said, disorders of epigenetic machinery, which Kleefscher is one. I'm going to talk a little bit about what that means, again, without going into too much of the weeds of molecular mechanisms. And my primary area of interest is in clinical trial readiness in these disorders and thinking about how some of this clinical trial readiness work can transfer between different disorders and be shared among these disorders. So you just heard from Sid, who I've known for a very long time, who does lots of work specifically to Kleefscher syndrome and thinking about these clinical trial readiness areas in Kleefscher syndrome. And I'm going to talk to you about some other disorders and how this may or may not cross over to Kleefscher syndrome, but kind of what can be accomplished through this clinical trial readiness work. Um, so I will share my slides. Okay.
Yep,
Um,
coming through great.
Perfect. So these are my disclosures. Um, And so we're going to talk a little bit about the history of clinical trials in genetic neurodevelopmental disorders. We'll talk a little bit about Mendelian disorders of the epigenetic machinery. And then we'll talk about outcome measures and biomarkers for clinical trials. So just thinking about syndromes that involve genetic syndromes that involve intellectual disability. Prior to the 1970s, really what we had were just clinical descriptions of patients that led them to be grouped together into syndromes. So these syndromes were typically things that were really distinguishable based on facial features or other features that led them to be able to be clinically grouped together. And then the next kind of 30 years after that became the advent of genetic testing and the ability to discover specific genetic mutations that cause syndromes associated with intellectual disability. At this time, really, most of the work was still in just the discovery of specific genes, and there wasn't as much work into mechanisms, and it wasn't thought that these were at that time treatable. So then moving into this century, more people started to look into mechanisms. So, yes, we know that there is a gene mutation, and these gene mutations cause syndromes that have intellectual disability as a part of them, but how does that happen? What's actually happening between the gene and the brain? And people also started to think about, are there therapeutic options for these? And lots of different thoughts have come up about ways that you could potentially treat these different genetic intellectual disability syndromes. And then moving into the present, there are some actual clinical trials that have begun because people did the work to look at the mechanisms underlying and therefore developed potential treatments. However, this has not been all good. This has come with some pitfalls as well. So, two prime examples. In 2011, 2012, a drug called arbaclofen had really promising clinical data and entered phase 3 trials for a genetic intellectual disability syndrome known as Fragile X. The arbaclofen trial used caregiver reported scales only as the primary outcome measure. And what happened was they did this whole big expensive trial where investigators and patients dedicated themselves to this trial, and the trial failed. So basically, they didn't see an effect in the group treated with arbaclofen. So, bummer, but that happens, right? It's things just don't work, even though you expect them to do so. The thing that's an even bigger bummer about the arbaclofen trial is that really, even to this day, no one, including the investigators who ran the trial, feel confident that they know whether or not arbaclofen actually works for certain things in Fragile X syndrome, because what they think is they didn't choose the right outcome measures. So they didn't choose the right way to measure whether or not these treatments worked. Because if you come to me and I give your child a treatment for their Kleefstra syndrome, I'm not going to just send you home and then in 6 months call you and say, hey, so are they better? What does that mean, right? What is better? What specific things are we looking to be better? How much better is better enough? These are all the things we need to know when we're designing outcome measures and doing this clinical trial readiness work. And so in this trial, they saw effects in some areas, but the trial wasn't designed to specifically measure those areas. And so what happened is these trials didn't work, and we still don't know whether or not arbaclofen could be beneficial for these patients, and that therapy has been lost. And in 2023, so much more recent, right? It would be nice if we totally learned our lesson, but one big pharmaceutical company, Roche, suspended the development of a ASO, which is a type of therapy for Angelman syndrome, after their early phase trial. They used really broad cognitive and behavioral outcomes, and they didn't see an effect in this primary outcome, although they saw a possible effect in an EEG biomarker. And so, again, what all this teaches us is a few things. One, you need to be really, really careful when you are picking your outcome measures. You don't want them to be too broad because you might miss your effect. And you want them to be targeted to the disease you're interested in treating. So, if we were to do a clinical trial in Kleefstra syndrome, we really want to measure the things that are specifically problematic in Kleefstra syndrome and the things that are specifically measurable in those problematic areas and the things that are clinically meaningful. So, there are lots of things you could measure. I can measure how many ankle sprains a kid has throughout the course of the trial. But at the end of the day, that's not really clinically meaningful to families of kids with Kleefstra syndrome. So we need very good outcome measures. And we also need these things called biomarkers. And biomarkers are things we're going to talk more about. They go along with the outcome of interest and they help us decide who's going to get what therapy, or sometimes they help us decide, is the therapy working earlier or in a different way than the outcome measures. And so my goal is that for the disorders of the epigenetic machinery, that we don't have these problems that I'm listing here, that we really do the clinical trial readiness work so that once the trials are launched, what the outcome that we get, whether they're successful or unsuccessful trials, that at least we feel confident that we know that that is the answer, that if they were unsuccessful, that it's just that the drug or therapy was not correct. And if they are successful, that we really measured something that was meaningful. So these are the things you need to really have a well-designed clinical trial. You need a specific patient population, a therapy that's being tested, and appropriate and clinically relevant outcome measures and biomarkers. So how do we get there? So we need to do this work that we call clinical trial readiness work. And these are kind of four of the main aspects of this clinical trial readiness work. We need to have a deep understanding of the phenotype or the symptoms that is specific to each disorder. So it's not enough to just say intellectual disability. We need to know, how is this child actually performing? What are the specific areas of strength and weakness? And how kind of bad is each one or good is each one? We need to have a deep understanding of the underlying mechanisms and downstream targets, so that we can understand the possible overlap between disorders and possible common treatments. We need to look at all the work that our basic scientists or preclinical scientists are doing and make sure that that work that they're doing really applies to humans. So it's great to have animal models and cellular models, and we can't do anything without them. We just need to make sure that the work that we're seeing actually also will be translatable to humans. And then we need, as we've been talking about, valid and specific biomarkers and outcome measures for individual disorders. So what all of the things that we're talking about here, Kleefstra included, are epigenetic disorders. So these are ways in which genes can be made into proteins, and you can affect how these genes are made into proteins without affecting the DNA sequence. So the EHMT1 gene or protein, what it actually does is turns on and turns off different genes and makes those genes more and less available to be made into proteins. And that's why we call it an epigenetic disorder. Um, and so more specifically, we call it a disorder of the epigenetic machinery. So again, EHMT1 is a protein that places a mark on these things called histones, and then that makes genes more or less available for being made into proteins, and then proteins carry out all of the functions in the cell. And so Kleefstra syndrome is a disorder of the epigenetic machinery. And something that we know, and the reason that I am so involved with these disorders, is that we know that the epigenetic machinery proteins have a huge role in cognition and behavior. So they are important to many things, but they're really, really important in the brain. And so all of these disorders of the epigenetic machinery individually are rare, but together they actually account for a large portion of genetic intellectual disability because all of these epigenetic machinery proteins are so important in the brain. And beyond what we know this from lots of other studies, both not only in the specific rare disorders of the epigenetic machinery, but also just looking at the basic science. We know that chromatin remodelers, whether that, which basically are things that change the way that genes get made into proteins, as I've said, are really essential for neurons to form and go to the right place that they are supposed to go. They're really important for the glia, which are the cells that surround the neurons, to go to the right place, to develop, to become the cells they're supposed to become, and go where they're supposed to go. And in addition to that, we also know from studies looking at other things that are not specific genetic disorders like depression, schizophrenia, that these histone modifications are really important for behavior. And when we ask you all, the families, what's most important to you all about your child's thing, you say aspects of neurologic functioning, right? So you say cognition, you say behavior, you say sleep. These things are all really, really important for and our functions of brain function. So that's really what we want to look at if we're looking at clinical trials in Kleefstra syndrome or pretty much any of these disorders of the epigenetic machinery. So we can divide these disorders of the epigenetic machinery into different categories based on their function. And what we're going to be talking about today is a bunch of writer disorders because Kleefstra syndrome is also a writer disorder. So EHMT1 places a mark on histones that then carries out gene expression functions. And so when you lose one copy of EHMT1, you don't have as much of that mark and it causes problems. And here we're going to be talking about other writers that, when lost, cause problems. And one reason we are talking about all of this is because it is possible that these things are treatable, right? It is possible that we can treat these epigenetic disorders through epigenetic modification as well as the traditional gene therapy and other ways. And so it's really, really important that we get the populations ready for clinical trials because mechanistically it's very possible that we can have some of these soon. So another writer disorder of the epigenetic machinery is Kawuki syndrome. Also, Rubinstein-Taybi syndrome is a different writer, and Wiedemann-Steiner syndrome. So those are the three disorders, all related to Kleefstra syndrome, and that they're all disorders of the epigenetic machinery, and they're all writer disorders. And we are going to talk about these three disorders in this talk now and talk about how clinical trial readiness has advanced in these three. So the first thing we're going to talk about is how deep phenotyping of these disorders can lead to outcome measures. So using Kabuki syndrome as an example, we looked in depth at the profile of Kabuki syndrome. And because again, it's really not enough and it's going to not be specific enough and not lead to good clinical trials to just say intellectual disability. We need to know more of what specifically is going on. And so we looked at a bunch of individuals with Kabuki syndrome, some individuals with intellectual disability who didn't have Kabuki syndrome, and then compared them to typically developing individuals. And what we see is that Kabuki syndrome have these specific abnormalities in visual motor, memory, and perception compared to their language skills and their general cognition, which are actually much stronger than those visual spatial skills in individuals with Kabuki syndrome. This kind of work allows us to then say, okay, so cognitively in a clinical trial, we are going to target these visuospatial types of outcomes. And so now, so the next step is to do work to specifically pick which subtests are we going to do? How are we going to score these to make sure that we're measuring these specific functions in Kabuki syndrome? But for example, if we did what we've done in the past and done broad measures, we could have missed the mark here because kids with Kabuki syndrome are pretty good in their language skills. And therefore, if we measured that, we're unlikely to see that much of a difference. So this deep phenotyping is really important. Deep phenotyping doesn't only have to be done in cognition, it can also be done as far as the behavioral phenotype of these disorders. So when we looked at Kabuki syndrome, they have pretty low rates overall of maladaptive behavior, but their anxiety is really, really high. And so, again, if we're looking to design outcome measures for clinical trials, what this tells us is that anxiety is something we really want to target. And going beyond this, we can also see that anxiety increases in individuals with Kabuki syndrome as they get older, which leads us to say, okay, so even as we're thinking of designing specific outcome measure batteries for different ages for clinical trials, we know that the older the participant, the more important the anxiety measures are going to be for measuring a response to therapies. Then, when we looked at other behavioral phenotypes, we saw that the ADHD phenotype is the most prominent behavioral phenotype in the very young children before the anxiety phenotype has become really prevalent at that point in time. And so, again, that gives us another specific target to say, okay, if we're looking at clinical trials in a really young age group, we're actually probably not going to look at anxiety as the primary behavioral in that age group. For this age group, it's going to be the ADHD symptoms that are going to be the more prevalent and that we're more going to be able to measure treatment effect. So once we have this data on this, we can think about other epigenetic machinery disorders. So, for example, another syndrome called Wiedemann-Steiner syndrome actually is a writer at the same histone mark as Kabuki syndrome is. And so we can ask the question, do these disorders have similarities? And what we see is, yes, they do. So Wiedemann-Steiner, as you could see on the left, like the Kabuki syndrome you saw earlier on the right, also have really specific visuospatial cognition issues with relative verbal language sparing. And so again, this is a unique phenotype that really crosses over between these two disorders that also have shared molecular mechanism, allowing us to say we can potentially do a clinical trial in both of these disorders and use some overlapping outcome measures because these are similar. And given the similarities in molecular mechanisms, it's also theoretically possible that we could even use a similar treatment. In addition, looking at those behavioral measures, both have a lot of ADHD early on and lots of anxiety later, but they have a very pro-social nature. So again, this is where this deep phenotyping is important because had we measured some autism characteristics in these individuals, we probably wouldn't have seen an effect because in fact, while they have some autistic features, often related to anxiety and other things, they are extremely pro-social, and so certain autism measures would miss the mark. So moving beyond deep phenotyping, we can also look at molecular and neurologic mechanisms to potentially lead us towards development of biomarkers. So one thing we know is about this concept of long-term potentiation. So long-term potentiation is basically a way in which neurons, after they have seen a stimulus multiple times, actually react differently to that stimulus in the future. And, this concept is called plasticity, where you can change the way neurons react. And it's thought to be, in certain neurons, a mechanism of how the brain actually carries out learning and memory. And so, in this other disorder of epigenetic machinery, a group showed more than 20 years ago now that, in fact, mouse models of Rubinstein-Tabes show deficient long-term potentiation. So they are not actually kind of learning as much from the electrical stimuli applied to the brain as they should. And so that could potentially explain some of the cognitive dysfunction in Rubinstein-Tabes. So given what we know about the way that there are molecular similarities between Rubinstein-Taybi syndrome and Kabuki syndrome, we decided to ask the question um, that one of my graduate students performed, asking, do the Kabuki syndrome mice also show the same pattern? And the answer is yes, they have disrupted neural plasticity. So they have an increased signal in the early phase of long-term potentiation, where the Rubinstein-Taybi mice showed no signal there, and they have decreased long-term potentiation, late phase long-term potentiation, just like the Rubinstein-Taybi mice. And I'm gonna, in the interest of time, skip on to the biomarker development. So then we asked the question, can we use this long-term potentiation to target biomarkers and develop them for multiple of these disorders of epigenetic machinery? So again, a biomarker is an objective and quantifiable characteristic of biological processes that is observed from outside of the patient, and it can be measured accurately and reproducibly. There are lots of different kinds of biomarkers, as I've mentioned before. Some we call like predictive biomarkers, and these may be, hey, this group of patients or this group of patients is going to probably respond better to this therapy or that therapy. There can be prognostic biomarkers that say, okay, we know your child has Kleistrer syndrome, but there's definitely variability within the population of Kleistrer syndrome as far as cognitive outcomes, etc. And so can we predict where that child's going to end up? And then there are things we call response biomarkers, which can use to be measure a response to a therapy. So these kind of go along with the outcome measures in a clinical trial. And it greatly increases the chance of a clinical trial being successful when you have biomarkers as a part of the clinical trial. So we have been looking at an auditory evoked potential biomarker. So this is done with an EEG, and it is looking at the way the brain responds to certain sounds that are played, and it reflects kind of cognitive processes, but most importantly, the way that we are measuring these ERPs, it can approximate long-term potentiation in living humans. So we obviously cannot cut any humans' brains and take out cells and test them in a dish the way we do for the animal models. So this is a way that we can measure that long-term potentiation that you saw was deficient in multiple animal models of multiple epigenetic machinery disorders and measure it in human beings. Um, and so we have done this in a number of patients, about 47 individuals with Kabuki syndrome and about 20 individuals with Rubinstein-Taybi and 15 with Wiedemann-Steiner, in addition to about 50 typically developing controls. And we looked at these sort of evoked potential parameters. So we looked at these change from the pre block to the post block. So we played the stimulus multiple times, and we're looking at how basically the brain is learning from these beeps that we're hearing. And what we see is that for a number of these pre to post parameters, so these are the long-term potentiation like parameters in all three of the syndromes, that we see significant differences from typically developing controls. So what this is telling us is that when we look at these ERPs, we can predict what group you'll fall in. So whether you have KS, RTS, WSS, or a typically developing control, based on these auditory evoked potential parameters. which is really, really exciting. And so our next goal here to further develop this as a biomarker is we've also collected a great deal of cognitive and behavioral data in these groups of individuals, and we're going to continue to collect more in each group and look and see whether or not these differences in the AEPs or the auditory evoked potentials actually correlate with cognitive differences in these individuals and behavioral differences. So, in conclusion, we could say that clinical trial readiness needs to be improved in to better have better, more successful future trials in genetic neurodevelopmental disability syndromes. That we need to characterize and understand the individual features of each of these disorders of epigenetic machinery in order to develop the biomarkers and outcome measures for clinical trials. And that clinical trial readiness research leads to these specific outcome measures and biomarkers. And that in fact, when we do this research, it's possible that some features of clinical trials, such as biomarkers and outcome measures, may cross over between different disorders of the epigenetic machinery, which then makes these individual disorders a lot more attractive for funders, pharma companies, et cetera, to pursue clinical trials in these. Um, and that is what I have. These are just my references and thank you slides, and I will take questions.
Thanks, Jackie. That was really on point and really interesting. I think it really fits in nicely with a lot of the discussions we've had earlier today. Um, just one quick point for those watching Jackie's slides. I think most people notice when Jackie has KS, she means Kabuki syndrome. Because
Sorry.
or KS this, KS that, and we're talking about Kleistrer syndrome. Unfortunate acronym alignment, right? But I just
Yes.
want to make sure no one got confused that when she was showing KS examples in her slide, she was talking about Kabuki syndrome, which is again is another writer disorder, and so we expect maybe has some potential similarity. So very, very interesting. Thank you. There is a question. I'll just read it for you from the audience. Anxiety seems to be a common issue in Kleistrer syndrome. Oh, that's definitely true. Clinically, have you treated patients with Kabuki syndrome or Kleistrer syndrome with a medication such as buspirone, which specifically targets anxiety, but seems to have fallen out of favor in the last decade? If so, what's your experience been? Is this useful in NDDs, or what were your thoughts on that medication?
Yeah, so it's a great question, and I didn't show all of the anxiety data that we have in all of the syndromes, but it does seem like at least in this subset of kind of writer disorders that have some overlapping molecular mechanisms, that anxiety seems to be kind of the predominant behavioral feature, and we see it over and over and over again, which is just a really intriguing research question as to what are these epigenetic mechanisms doing in the brain that's causing this very specific behavioral feature. And so the answer is yes, I treat lots of patients in clinic for anxiety with lots of different things. I do not tend to go to buspirone first line. Because there's better that while there's not any specific evidence in any of these disorders yet, there's better evidence for some of the mechanisms that I believe may be underlying the anxiety for some other drugs. So I tend to use like SSRIs kind of first line because those are known to increase neurogenesis in the hippocampus, which we think is maybe a mechanism behind some of this epigenetic dysfunction causing anxiety. And then I often also go to mood stabilizers because those may have some molecular effects that also treat the underlying anxiety. But it's a really, really important question. And I'm glad you brought it up because uh, yesterday I submitted a letter of intent to a PCORI grant
Mm,
asking them to fund looking at anxiety treatments in these epigenetic machinery disorders, not to like treat the underlying, not what we call disease modifying therapy, but just to really be able to answer your question, which I get all the time from families, which
mm,
is what should we treat the anxiety with? We need help with this.
mm. Okay. One other housekeeping item is a request from the interpreters to try to speak a bit slower. So I am
Oh,
one of
sorry.
the most guilty. No, I think they're mostly probably talking about me, actually. I'm one of the most guilty people. So I will try to slow down a little bit. So I had a question for you, Jacqui. So this is probably true in Kabuki as well, and the other syndromes you've studied. In Kleefstra syndrome, the phenotypic variability is very broad, right? So we have people who are very high functioning and people who have a lot of real challenges. So people who walk really well versus people who have no mobility, almost no mobility. People who are verbal versus people who are completely nonverbal. How does that affect when you're trying to find these standout um, phenotypes, right? That really are different between Kabuki versus the regular population.
Mhm.
How does that variability come into play? Where maybe someone who's very high functioning, how would you fit them into that? Does that make it challenging to find these? I guess is my question. And how do you deal with that? Because I'm assuming Kabuki probably has the same kind of broad variability.
Yeah, so it does make it challenging. Of the three disorders that I presented, it's probably Rubinstein-Taybi syndrome that is the most similar to Kleefstra syndrome as far as the variability, where you really get the entire range of functional abilities. And so, that's why we try to do as many patients as we can so that we can really hone in on the phenotypes. And we, in some ways, compare each patient to themselves, if that makes
Yeah.
sense. So meaning
Hmm.
we want to not only see how do they compare to the general population, because that's kind of less interesting. What we really want to know is when we look at all the different domains in any of these disorders, do we see a consistent spread, even though some may be overall moved higher and some may be overall moved lower? And we really hone in on that. So for Kabuki syndrome, yes, we have a range of visuospatial abilities, right? Some kids are, you know, 2 standard deviations below the mean. Some kids are 5 standard deviations
Hmm.
below the
Hmm.
mean, right? But they all, pretty much all of them, when you look at their overall abilities, it's less than their language abilities. Whereas if you look at, say, their executive function for Kabuki syndrome, right? For Wiedemann-Steiner syndrome, it's different. If you look at their executive functioning, it's all over the map. So we say, okay, executive functioning, not what we want to target because it's
Hmm.
way too variable. But visuospatial, we can measure this because there is always this deficiency. So
Right.
those are the kind of things that we want to target and look at. And then the other way that we do this is all of the biomarkers that I show you, I'm showing you some early data where we're comparing it to typically developing controls. But the next step is to really understand how these biomarkers relate to the cognitive and behavioral phenotypes of the patients so that we could say, why is this difference happening? Why are some people with Kleefstra syndrome functioning this way and others this way?
The other question I had was FDA is really focused on patient-focused drug development, and that the way you measure if something works should be tied to what's important, what people say is important, right? So you have a disease concept model, and these are the things that are important to the community. Maybe that's sleep, maybe that's cognition, maybe that's behaviors, et cetera. However, what if cognition is not something that has a good differentiation, right? When you're looking at your measures, how do you find a way to sort of thread the needle where you find measures that both are important and you can say we have a very strong differentiation here and a very strong signal that we can capture? Or maybe is it not that hard after all at the end of the day?
It
I guess
can
that's my question.
it can be hard to, as you say, thread the needle. And so that's where I'm really interested in not looking at super broad measures like, say,
Yeah.
cognition and saying, okay, the community says they'd like that, they'd like cognition or school performance or whatever it is to get better, right? Okay, but what are the specific things within that we
Yeah.
found for Kleefstra syndrome, say, and how can we measure that specific
Yeah.
domain instead of
Right.
cognition more broadly? And hopefully, the goal of that is to, like you said, thread the needle to please both the FDA, uh,
Yeah.
make a valid clinical trial, and of course, make a clinically meaningful difference.
Yeah. And I had a question about the graphs you showed where you were finding subdomains, I guess we could call them, where there was very strong differentiation in, say, Kabuki syndrome. Were those based on the commonly available clinical assessment, just like Vineland and so on, and then you were taking sub slices of them? Is that how you were finding those parts that were very strong, or were you making totally new types of clinical assessments?
No, so exactly for the reason in your previous question, right? It The FDA doesn't love totally new
Yeah,
assessments, right?
right.
They that's not their thing. And so it can take a very long time to get to clinically trial ready for a brand new assessment. So
Yeah.
typically what we do is we exactly what you said. We take a measure and we slice it up, right?
Yeah.
And look at this specific subtest or this specific domain. So, in order to compare to general cognition, let's say we do a Wechsler measure, for example,
Mm.
in an individual with
Hmm.
Kabuki syndrome, where we do the whole IQ battery, but then for the primary outcome, we only pull out block design, let's say, right? We
Okay.
use
Yeah.
the whole IQ test just to give patient demographics, if you will, so we know where they are. And then we pull out the specific subtest, and then we create sometimes, which is still a new measure, but the FDA is usually more comfortable with it if you can provide data. Like for Kabuki syndrome, the FDA has approved a composite measure where we pull a subtest from here, from this test, a
Mm.
subtest from this test, a subtest from this test, and we use them
Yeah.
together.
Okay. Very interesting. And then we had a comment from the audience. So it's not really a question, but my son recently went on Abilify, and I'm floored at how much has it improved irritability and dysregulation. So I don't know if you have any comments on that patient experience.
Yeah, I mean, I think that this question of, medication to target certain symptoms is incredibly important and understudied. And there's probably a lot of things out there that have both symptomatic effects as well as some disease modifying effects, right? Lots of things actually have epigenetic effects that we don't know
Yeah.
about. And so some of the questions we may need to be asking is, are there combinations of things that already exist, like a little bit of this and a little bit of this and a little bit of this,
Yeah.
that can really make a big difference in some of these things? And the goal of clinical trial readiness research, which, Dr. Sid was just presenting and that I've presented here, is to say, okay, so whatever we want to measure, right? Whatever we want to test, therapy wise, we have a clinical trial to do that, right?
Yeah.
So it's not a one and done thing, but we have this clinical trial readiness for eternity.
Yeah. Absolutely. I think that's exactly where we want to be, and it's a really will be a really exciting place to be when we all get there. So thank you for all you've done. Thank you for presenting today.